Okay, we are almost there for the day. So we thought just to, according to your schedule, we have this in-vivo cancer models. Okay, I thought even though we have covered most of these cancer models, I think we should just go through and make sure that you know all those models, and also in each one what is the difference, right, and why you want to use it. I think it's important. Um, these in vivo models are essential for studying cancer biology and therapeutic efficacy and also metastasis in the whole organism. So you all know already in each one, like for example, tumor biology, cancer progression, you know what models we have used, genetic model or any model, cell model or anything. Therapeutic efficacy, you know already, orthotopic or xenograft and genetic mouse model, why we use it at what stage, what for. And you also know metastasis model, a little bit of it, not completely, but still you know the models, why we use it. And when they metastasize, and also the cancer has to be progressed to metastasize, right? And also these models, in vivo models, help in validating the in vitro findings. I think I mentioned yesterday that what in vitro findings you have found, and basically you're validating it. For example, I think I was telling someone that you have to see the colony formation assay and see what happens in the colony formation assay. If you have seen the result in colony formation assay, usually it mimics in the in vivo mostly. Maybe 90%, don't know, but it will usually mimic in vivo. So if you are doing it right there, it actually in vivo you get that result, unless until there is other technical issues or cells have an issue or anything else. So why we use these models? Because some of these models recapitulate the way the human biology or human cancer progression happens. That's why we use it. That's critical for advancing the translational, of the cancer progression or cancer research, improving also the clinical outcome of this, study, whatever that you are trying to establish or do. So the types of in vivo models, right? We have a xenograft models, we have a syngenic model, genetic mouse models that we talked about. We also talked about patient-derived xenografts, okay? And also we talked about organoids. Those are kind of, we actually derive from these xenografts. It's okay if you cannot do it. At least you have that knowledge of what models you have, we have, I know people have it. Then at least you'll know when to use it or how to use it, right, if we have that opportunity. So in the xenograft model, we implant human cell lines, right, or the tissues into the immunocompromised mice, right, because you are using the human cell lines, the mice are immune compromised, so you basically, you're just testing them, that's all. So you basically not mimicking the whole microenvironment or the whole thing that in how it is in the human, in the mice. Then you use a syngenic mice model where you implant the murine cancer cells, like mice cancer cells, back to the immunocompetent mice, where you basically get the immunity, whatever the microenvironment in those mice. So most importantly, the engineered mice, okay, the spontaneous tumors are fantastic because it has everything, how you see in the human, right? Probably human microenvironment, all other things. That's why, that's the reason that people try to establish in the genetically engineered mouse model something if they want to study either biology or therapeutically, right? Patient-derived xenografts are very important nowadays. If you think the technology is so much developed, but you all know probably that the patient will be lying on the bed and they take the tumor and we will be working in the laboratory. For example, our own laboratory, what we do is we take the patient tumor and we divide the tumor and we study everything. Like, one person will be studying something, one person will be injecting, cutting those tumors and putting into the mice, orthotopically and growing them, and then again they take and then cut, and again they grow in more mice, and then therapeutically they'll be doing it. The patient will be lying. They'll be going through the chemotherapy after the surgery, right? And we do the lot of chemotherapeutic agents and combination whatever we think is promising. And if we see any great result, probably we will take that result back to the clinic and tell them that this is what I think this patient tissue is responding because of for this kind of drug. I'm just giving you a hint, kind of that this kind of studies are gone in many laboratories and cancer centers in all over. So it's also important. I mean, your cancer centers has to have the approval to conduct this kind of studies. Otherwise, not all the cancer centers can do. Okay. And when you take the xenograft models, there are cell line derived xenograft models. Okay, there are advantages where you can establish a tumor, and there is a reproducibility. Okay, is easy. There is a limitation, lack of tumor heterogeneity. Okay, you don't see the heterogeneity in the cell line derived tumor. For example, I was telling you about the human cell line. You take and there is no heterogeneity there. Okay, you just study drug. Okay, the tumor going down. That's it. That's why if you submit any paper, probably I don't know who has any experience, but if you submit a paper, reviewers ask, okay, there is no tumor microenvironment in this. Only you're just showing the tumor reduction. That's not enough. Okay. So the patient-derived xenografts, it's better, it's preserved, tumor architecture, heterogeneity, and genetic profile is there because you're taking the patient tumor and genetic, whatever the genetic is, profiling in the tumor, it's still there. But the problem in this study is that you need the immunocompromised host variable, so that's expensive. You have to make the mice with immune, and then you have to study the human tissue in those immune, because you have to develop those kind of mice. It's like very expensive. Okay, so I showed you one of the data. First talk, I showed you humanized mice we developed, and then we studied the CREB inhibitor in those because our study took us there, and we did study those mice, but one mice is $3,000. Okay, just imagine, convert into rupees, like how expensive it is, just one mice. So like that, you have to have multiple mice to study. So it's a little bit expensive stuff. Okay, when you take the syngenic models, right, you take the murine cancer cell lines and implant into genetically identical mice. So the advantage is good. It has an intact immune system and allows the immunotherapy studies you can do. I showed you one of the immunotherapy study that what we did, and that's exactly what I'm trying to tell you. Advantage, fast, effective, things like that. But there is any model you take, there is always a limitation. These models have, they don't recapitulate the human tumor biology. True. It's like, there is no 100% perfect human, model that you can actually study. It's all, but these models will give you some kind of, data. That's why you go to the phase 1 and study the toxicity and other things. But it's okay. But important for you is that when you're doing the research or conducting the experiments, I think thinking all about these things and conducting whatever you can, right model that based on what you are thinking, if you can do. If you cannot do, there's always a collaboration. If you cannot do collaboration, just whatever you have, publish it, right? Wherever you can, right? Don't waste those data if that is reliable, good data, okay? So when you take a genetically engineered mouse model, it doesn't matter which cancer. We already discussed that you have a genetically engineered mouse model. They spontaneously develop tumors, and also it relapse and resistance and all those things. We can study a lot of things. There's like you can basic cancer research you can actually study. I told you what we can study is that you take the we did that. We have a KRAS mutant mice model. We have a KRAS and also P53 mutant mice model. In the KRAS P53 mutant mice model, it actually develops cancer. KRAS don't develop cancer, but it has KRAS mutation. But it needs another thing. That's why we did the alcohol and induce other things. So we made the cancer. But here we do the cancer research, like for example, study the cancer progression. You can use those models. Also, we can do therapeutic. You use those models and study because it recapitulates exactly how the human biology and cancer progression happens, right? That's why there is an advantage and disadvantage. There are more advantages than the disadvantages, but it's just that how why you want to study in genetically engineered mouse model is a key factor, right? The limitations are the longer latency periods, higher costs. Genetic mouse model is not easy because you have to maintain the facility and maintain the mice. I don't know whether anybody is working on genetic mice model. It's hard because for the for facility you have to have in the universities or institutions. But those who are having it, definitely there is great papers are coming out from their laboratory. And the complexity in breeding and genetic you have to have the genetic person all the time with you. Because I have a colleague called Michael Vanson. He's always with me, even from Vanderbilt University to now. Now we are a postdoc together, and we went to a faculty, and now he's associate professor in somewhere else, and now I'm in some other university. But he's a geneticist all the time. Anything genetic problem, I'm not a geneticist, right? So basically he is the one. So any mice model or anything like that, I always ask questions, and he's my collaborator. We publish together and we develop anything ideas, and he makes the model and I actually use it, and sometimes we collaborate each other and makes the model. Like, if I have scientific questions and I discuss with him and make those models, and we figure it out, and that's how we develop the relationship in the scientific world and collaborate each other and develop. He's from different Kansas University, I'm in Miami University, it's still like that. What I'm saying is that you have to have someone who understands the genetics of these mice, and the crossing, breeding and all those things. It's not that everybody, you cannot think like, I can start doing it, but you have to have that basic knowledge, and also understanding the genetics, which is different. Okay, we have only, if I am from biochemistry, I studied the biochemistry, a little bit of genetics, a little bit of molecular biology, a little bit of microbiology, immunology, it's like that, but not in depth anything, right? So that's why you need to have the genetics person. So if you are developing a mouse model, it is a zoology, like where they actually understand the chromosomal mice and those kind of things. They can actually collaborate with zoology people, those who are expert in genetics, something like that. So when to consider the model selection, right? There's a great website here, Cancer Model Database from the Charles River. And you can actually go there and check each model that you want to study, and they'll give you all the details about each model, what these models are, and like that. There are even NIH website they have it, all the details about models. But what I'm saying, even the Jackson laboratory, they have the mice model and the details. So like that, there are you have to spend time to figure it out, all these models, and if you want to. Like, if you don't want to, it's okay. But at least you get a knowledge. You can talk about it and you can discuss with someone and get a knowledge of the models, right? Does the model mimic human disease features? There is a question. You have to always have that, to relate your biology to your model. And also there are like an immune competence, whether, immune system. Do you want immune system in your mice model? Otherwise, why you spend so much of money? You don't have to. Think about your model, think what you are working on, and then you think about whether you want immune system or not. I gave you an example. In our study, we wanted immune system because it did not work MEK and STAT3. And then we looked at all the immune population. Then we found that, yes, we need the immunotherapy. Then only we went for immune, related studies on genetic mouse model and immunocompetent, immunized mice model, things like that. So it all depends on your scientific question and also what type of data you are getting it from your research. Tumor heterogeneity, again, you have to think about, do you want to study heterogeneity in your model? Why? If you're just studying the natural compound, just study the way that my first paper was, that we did not do much, okay? But study and see what's going on. And then you move on we moved on to the immunotherapy later on. Like we took like 9 years to 8 years to think about it and see. Meanwhile, we published other papers. What I'm saying is that it takes time. Also, you have to have the money and all those things. So you have to think accordingly. Okay. The cost, practical consideration, cost, time, and technical feasibility, whether you have that expertise and things like that. Okay. Experimental design consideration. So controls, replicates, how many mice you want to have. Now, let me tell you, 3-5 mice is not enough if I want to publish in Cancer Research Journal. Cancer Research Journal I published before in like 15 years ago, and they were fine. 3 mice I published it, and now no, it's not enough. So now they want more. 10 is much 10 is at least minimum sometimes because of the data, that what type of data you are generating. You know what I'm saying? So it's all about that. So the control, because the replicates, the reproducibility issue, like if you have 3 mice and you're like that, like whatever is happening, and if someone wants to reproduce your data, it's going to be difficult, right? I think that's the reason that they are asking more mice, right? Important. It's just, even though they don't ask you, they reject your paper. They won't tell you why they are rejecting also specifically, right? There may be reasons for that, okay? One of these may be the mice number. The endpoint selection, what are you trying to establish in your study? Only tumor growth, metastasis, survival? Those kind of questions are also important when you are designing your, in vivo models, okay? Based on that, you have to design your in vivo models. You don't have to go, design or what you want to do, I want to design. There should be your question or your data should take you there. And also ethical consideration, animal welfare guidelines, and also minimizing the suffering. So there is always a guideline for everything. Probably for the human patient, you can actually cut and open, easily. But mice, you cannot do it. You have a lot of restrictions. I don't know here, but in US, you cannot just handle the mice like how you handle human. Human is easy compared to mice. Let me tell you, there is a lot of rules and regulations. Even to each mice that I have to sack it, I have to have the regulation, each and everything, how exactly I'm going to sack it, whether I'm giving the pain to the mice or not, and how long I'm doing the drug treatment, what food and whether the light is there or not, whether they are sleeping or not, whether they are active or not, whether they are having any problem or not. I don't think we care about so many things like in the OR also in the surgery. I don't think so. So it's like that. So we have to take care of. Like for example, I got 3 emails from an animal person that something is happening in some of my mice that somebody has to go and take care immediately. You know what I'm saying? So it's like that. Anything happens to any mice, like immediately someone has to go and check, make sure they're fine. So it's like that. So what I'm saying is that ethical consideration as, you have to consider. So there are challenges and limitations. Of course, when you start conducting these experiments, you will know. Okay, it looks fine. When you see the data, it looks fantastic, everything. But we do go through a lot every day. Okay, as I said, ethical considerations are very strong ethical considerations in US. So basically, we have to go through a lot of things. The differences between the mice and the human biology can affect the translatability. Yes, not all your data, not all your research translatable. Not all. Okay, some fail. Many reasons. Each reason is important reason if you can figure it out. Okay, important for the scientific community. It doesn't matter what type of limitations or problems that you have. Each problem is important. Anything that you think your data is correct, that means it is important to the scientific community. So microenvironment, right? Any difficulties in replicating the human tumor microenvironment? Yes. You cannot replicate the tumor microenvironment from human to the mice to any other model that you think about. We think we are just conducting we just don't know a lot. Okay, we predict a lot, and maybe technology is there, but technology is there, we are using it, and we think that that is how it is working. Probably we might not know a lot. Okay, we might know a little bit of it, what's going on in these mice. Okay, that's how probably we also scientists say, in cell we understood only a little bit of it. There's so much that we don't understand. Okay, So like that. So the same way in the microenvironment, we don't understand so much. But important is that we don't have to worry about the negativity of what is the problem. We have to think about the positive. Like we have these excellent mouse models we should study. We should be able to establish some research and science, right? That's what you need to think about. Don't worry about the limitations a lot, but you need to know, okay? The cost and time for developing the GEM and the PDXs is a lot. You have to have a lot of money to do all these kind of studies. But if you have money, I know there are a lot of companies around in Bangalore. I know they make the PDXs GEM model from the companies, and I know there are some few companies came to ASER meetings also from here, from India. So also these companies are selling all these mice and the PDX. If you have money, I think you can actually get those mice and study. So the best practices, I mean, I think Joshi was talking about in vitro study. Here also in vivo, the standardized protocols are there, you have to follow, and then validating the model relevance to the human disease always. Why are you studying in vivo? Because you want to give the relevance to the human biology, human disease. That's why you're studying in vivo. So that's what I think you need to think about what you're validating is whether it's helping you, whether you're actually getting the data or not. So the future direction, I don't have to say future direction, but actually it is true that you need to develop the humanized mice model to incorporate the human immune component. It's important. Okay, when you're studying, you have to think about it. When you're writing a project, when you're proposing projects, right? Like you have to propose something innovative, something proposed, and see how much money is involved. Talk to company and get those codes and add it, and think about it whether you have the facility. If not, if you don't have it, attach to someone who has the facility. Maybe from here to Manipal. Manipal maybe having all the fantastic facility. Collaborate with them and try to establish your model system so that they'll fund you. They will give you the money. If your science is good, they will definitely fund you. If your science is like your thinking process is better than anybody else, definitely they'll fund you. It's just about your process. Okay, if you say you're not getting funded, that means that I will say your science, your thinking is weaker. I think you have to become stronger. You have to think beyond. That's why you're weaker. So you have to think how your science can be innovative, right? So the real-time monitoring of tumor dynamics, advancing imaging techniques like PET, CT. I've showed you some of those images, mass spec analysis. What I did not show, I showed you a little bit of those figures, MRI analysis. And what I did not show you is that the whole process of imaging technology, because that's another research and another area. We have done a lot of in vivo imaging, PET, CT, MRI, designing and even throwing to the mice and getting those data and seeing all this data, entirely different area. Okay, we did not touch those. Okay, multi-omics integration. So how you can increase? We already talked about genomics, proteomics, transcriptomics, why, what, and all those things. I think it's all about whether how you are doing in the in vivo model. In vivo model, like how you do the in vitro model, it is not easy. Again, in vivo, your data will be different. So you have to think a lot. There is a lot of research. That's why it takes a lot of many years. It's just not 1 year, 2 year, 3 year, 4 year. It takes years and years. The reason is that there is a lot to understand and study and things like that. So your whole life will be thinking about a lot of these technologies and ideas. Even you say something, right? Like your 10, 15 years will be gone for studying just for that. That's why scientists are lying somewhere in the corner and they keep doing what they want to do, but they don't care about anything else because that's what it is. That is the passion that develop, right? Take home message: it is all integrated, well-designed in vivo model strategy is critical for advancing the cancer research and improving the patient care. Very much. In vivo data is a key thing. You might have got a lot of in vitro data. What are you going to do with it, right? What are you going to do with it? You have to somewhere, like when you come to in vivo data system, in vivo data, again, there are a lot of problems. Yes, there are problems. There is always problem everywhere in the research, and when you go to, but you should not think about it. What you need to think about it, whether your science, you're translating it or not, if you are in the cancer biology, it all depends on your disease and what type of in a food technology, probably you think differently, right? Your ideas are different, and your improvement and what you the best food product or best something, and how you package it and how you sell it, how you design and packaging system, or freeze drying, or how did you dry, or whether you're keeping all the flavor, keeping all the protein intact or not, things like that. They're different, but it's the same thing. End of the day, we all talking about the technology. We are talking about system, and to keep going in the right direction, and making the right path. Okay. Thank you so much. If you have any one question, I can take, because I think it's the lunch time. You all hungry? Any one question? So if not, we can discuss during lunchtime. Okay. Thank you so much.